staff member here in the Gene Expression Center. And I'm here today to introduce the panel from BioLegend. So for our clients who are interested in both single-cell RNA sequencing and also single-cell protein expression, Lisa's been a really great resource to help them kind of figure out what antibodies they need for their project and also to work with them on any technical issues that might be involved with the actual sample preparation. So she's here today to talk in part about some kind of cool new advances in being able to look not only at cell surface protein expression, but also now at proteins within the cell. So with that, I'll turn it over to her, and thank you for being here either in person or on Zoom.
Thank you, Harrison. Thank you guys for coming. Appreciate you trudging through the snow to get here. So as Harrison mentioned, I'm Lisa. I was formerly Scientific Applications Manager, now a Software Solutions Manager. So we are trying to get somebody into my previous role. But for now, please contact me if you have questions about this. I will make sure they get to the right place and for help if I can. But yeah, as Harrison mentioned, I'm going to talk today about our TotalSeq portfolio, which is our solution for single-cell multi-omics studies. Okay, so this is just kind of an outline of what we're going to talk about today. I'm talking about the concepts here, the TotalSeq antibodies, single-cell RNA-seq, site-seq, cell hashing, our panel solutions, intracellular protocol, and a new kit that we just released last week, the TotalSeq Phenosite for single-cell protein analysis, and our Multi-Omics analysis software so that you can analyze your data that you generate with these fun tools. Okay, so to get things started. So what if, we have some kind of condition, a disease, a treatment, some sort of health problem, or, a study, something you're looking at in mice, and, we need to study that particular indication. So you can do things with mRNA, single-cell RNA-seq, but you have some limitations with that, challenges of RNA dropout and a divergence between RNA and protein expression because they're not always the same. Conversely, we have lots of different tools we can use to analyze proteins, like flow cytometry. Limitations here are that there's limited multiplexing capacity. Yes, now with advanced cytometers, we can do 40 plus parameters, although that does take quite a long time to develop. But you also don't get simultaneous RNA data. So things you have to consider, of course, for either of these are the sample you have, the time and cost limitations for either of those assays. But what if it was possible to analyze both your protein and your mRNA in single cells simultaneously? And so a few years back, at least 6 years back, I guess it was, maybe a little longer than that, the technique called site-seq, or cellular indexing of the transcriptome and epitopes by sequencing, was developed. And so this was initially done using antibodies with biotin and then streptavidin antibody barcodes, and comparisons to flow cytometry and site-seq were done at the time. So in this application, you would stain your cells in a similar fashion to flow cytometry before you do single-cell RNA-seq. And then what you're able to do is sequence the proteins that are in your sample based on these antibody-derived tags, or ADTs. And so when you have your sequencing, you both get cDNA for further library prep and then antibody oligo products for further library prep. So you'd have two separate libraries that you prepare. And so the single cell multi-omics approach of site-seq, okay, it was more than 6 years ago, sorry. So it was a little longer than that. We developed, our products went online in 2018. So in the New York Genome Center in 2016, they integrated the protein information into that single-cell RNA-seq workflow, like I said, using biotin, streptavidin interaction to link those oligos to antibodies and then ran them in single-cell RNA-seq. And they correlated this method with flow and mass cytometry data, and this approach is compatible with 10x genomics. And so for a while now, we've been selling products in this arena. And so our antibody oligo design elements are as follows. So we have 3 main components in this oligo. So we have a PCR handle, which is Illumina sequencing compatible. We have an antibody barcode that's unique to each individual clone in our portfolio. And then we have a capture sequence that is modified for different formats of our TotalSeq antibodies. And I'll go through the different ones that we have in a couple slides. But basically, you can seamlessly integrate this with different single cell and spatial solutions based on the format that we have. Again, I'll describe in a little bit. And it's a pretty simple cell staining procedure that's similar to flow cytometry or IHC, depending on the application you're doing. And these antibody-derived tags, or ADTs, are captured pretty similarly to mRNA. And then at that point, we have an independent ADT library that is constructed for sequencing, and this is called your protein library. Okay, and so the nice thing about using oligos to indicate what clones we have is that there's a lot of unique barcodes that we can use. So in comparison to flow cytometry, where we're limited by the number of fluorescent proteins or fluors that are available to tag our antibodies with, we're almost unlimited at the number of barcodes. So your limitation here is the number of clones, really. And then we can choose a lot of different options for the barcode that's attached. What I will also say is that what we have in catalog, you don't have to be choosing the barcode. They're already selected, conjugated, you buy them, they're ready to go with the thing selected, and we'll tell you exactly what that 15 base pair barcode is for each clone. Okay, so the workflow looks a bit like this. So the first thing we would do is we would take our nice healthy cells, and we're going to label them with these antibodies. So we do antibody binding. If you are doing flow cytometry sorting to select a population of cells or magnetic depletion, there are ways to which you can strategize when you're going to do the oligo conjugated antibody staining. And so that's something that I would encourage you to come to us to talk about before you proceed with your workflow. You wash the cells really well. We want to make sure any unbound antibody goes away, because if it gets captured in this oil droplet, it'll get sequenced as if it were on the cell. We don't want that, okay? So we need to make sure that their cells are washed really well. And then we're going to proceed to this, in this case, this looks like 10x workflow, single cell droplet encapsulation. So we have cells, we have oil, they're going to go together with the gel bead to make this single cell. So one cell, one bead, and there's all kinds of different antibodies on that particular cell. So in the droplet, the cells will lyse, and then you'll have the mRNAs that'll start to bind the gel bead. You'll also have the oligo tag from the antibodies bind the gel bead. And then those mRNAs and antibody oligos are going to hybridize to the RT oligos and are indexed with a cell barcode. So then we start applying different components of the address, if you will, to tell us where exactly which cell the different antibodies and mRNAs came from. So after that process is complete, you would separate the antibody oligo products from the cDNA by size selection using spry beads. So then we have our gene expression library and our cell surface protein library, and they're separate. They have different sizes. This is all stuff that Harrison and team would do when they're doing the library prep. But that component's very important not to throw away your cell surface protein library, because it is in the supernatant of that initial selection. The libraries are then amplified and then pooled for sequencing. So basically, if you're doing the lab component and cell prep, you're kind of just up here, and everything else on here is done by the lovely folks at the GEC here. Okay, so what is the power of TotalSeq? So we can separate transcriptomically similar cell populations. So if you see here in these UMAP plots, we have just basing cells by what kind of cells we have in the mixture based on RNA, and you can see that there's kind of a big cluster of these different cell types here. When we add in protein to the mix, we're able to really differentiate some of these, what would be maybe smaller cell populations from within a heterogeneous mixture of cells, which allows us to get a little bit more detail on what we're looking at in a mixed cell population. So that helps you characterize cells by immunophenotype, and then also aids in identification of cells with low RNA content or poor RNA quality, if you have that protein as well. And so in this publication here, where they were looking at 25 different TotalSeq antibodies and on approximately 31,000 PBMCs, you can do different kinds of analyses like weighted nearest neighbor analysis to integrate multiple data sets together on the same sample to generate a single representation of multimodal data. So both the protein and RNA, I think up here, which is pretty cool. So some key takeaways on TotalSeq up to this point. So the antibody oligo tags are captured similarly to mRNA. You get an independent ADT, antibody-derived tag library. The ADT and cDNA libraries share the same cell barcode, and each oligo has a unique molecular identifier. So with all these components, we can tell what cell this belonged to and what antibody it belonged to, so we can figure out both mRNA and protein from the same cell. Okay, so I mentioned I would tell you more about our portfolio. So our TotalSeq reagents fall into well, formats is one thing, but we have different categories as well up here. So we have our primary antibodies, liquid vials, so single clones just on their own. And they come in different formats, compatible with poly-A capture platforms, 10x genomic supported products, and Mission Bio supported products. And so these are just the clone by itself. They're sold in 10 microgram format or vials. Then we have our total-seq hashtag antibodies or HTOs. We have 24 hashtags per format for mouse and for human. And so we have, again, poly-A capture platform compatible, 10x genomics compatible, and Mission Bio compatible. And then we have our antibody cocktails, which are pre-optimized cocktails containing 100 plus clones. And they've been pre-titrated and lyophilized so they can be reconstituted and used without having to do all the titration work. We have these in various formats, and the formats have differing numbers of clones, hence the range there. And we have those for mouse and for human as well. And then we have some secondary reagents. So streptavidins with or without dual labeled fluors, and anti-isotype specific and anti-tag specific, such as like anti-PE or anti-biotin antibodies, so that you know, if it really comes down to it and you can't get a conjugated version, maybe a secondary could be an option for you. So your protein of interest and there's no antibody commercially available, but you can biotinylate it and come in with a streptavidin oligo, or you can come in with a secondary to an isotype. Okay, so I mentioned we are going to talk about the different formats. So our portfolio contains 5 different formats of total-seq antibodies, total-seq A, B, C, D, and BN. Don't ask me why BN, but it's BN. So our total-seq A antibodies are platform independent. So because they have a poly-A capture sequence, anything that uses a poly-T is compatible here. So anything that's going to capture mRNA, theoretically. And so what happens here is we have our total-seq A antibody capture sequence, which is a poly-A, and then it's going to bind to poly-DT on whatever system that is. Then we have our total-seq B products, which are specifically compatible with 10x genomics single-cell 3' assays, whatever they're calling them these days, and they're flex gene expression assays, some of them. And they have a capture sequence 1, which is, the total-seq B antibody capture sequence is complementary to capture sequence 1 on the 10x products. Now, notably, you can use our total-seq A products with 10x 3' kits because they still have that poly-T for the mRNA. So those are still compatible. There's just different components you need to prepare the libraries at the end of the day. And then our total-seq C products are compatible with 10x genomics single-cell 5' assays and the flex gene expression, and also enable you to do immune profiling. So the clonality of TCR and BCR or VDJ with the 10x immune profiling kits. And those have a unique sequence that's complementary to the template switching oligo on the 10x genomics 5' assays. Then we have our total-seq D antibodies, which are compatible with Mission Bio for single-cell DNA and protein. You guys don't have one of those, do you? Okay, I think I might have heard rumblings about it, but anyways, so forget about those for now, unless you want to do some single-cell DNA, in which case we can certainly talk about it. And then our total-seq BN are compatible with the 10x genomics Visium spatial gene expression on FFPE samples. So for spatial transcriptomics, not going to talk about those today. They won't have time, but if you are interested in that, definitely ping us, please. Okay, so our single antibody conjugates, I'll talk about a little bit now. So they're in a liquid format. They're in PBS, page 7.2 with azide and 1 millimolar EDTA. We test all of our antibodies by flow cytometry to confirm that they bind the epitope they're supposed to, and Sanger sequencing for the construct of the oligo that's on there to make sure it's the right sequence. They are at they're all at the exact same concentration across the entire portfolio, and they come in 10 microgram vials, as I said. So they should be titrated for your specific application, and you can contact us for guidance on how to do those titrations and why you should do those titrations. Every single technical data sheet will say use at less than or equal to 1 microgram per million cells in 100 microliter staining volume and titrate for best performance. So when you're like, what does that mean? That's when you can come to us and we can talk about it. We have an entire blog on how to and a white paper even, I think, on how to titrate antibodies. So for this application, so we can definitely discuss. And the benefits here, we have the largest single antibody portfolio with oligos on them in the market. They're easy to use. They're pretty good test value. I say 10 micrograms, which realistically, people who do a lot of flow cytometry say, well, those are sold in 100 microgram format, but you're probably not doing the same kind of throughput assays when you compare those two. So 10 micrograms, really, if you're not using at 1 microgram, but you're using at 0.1, it's still going to get you 100 tests in the long run. So, and they can be pooled to make your own panels. So because we're looking at oligos here, we're not thinking about fluors and fluorescence overlap. You can pick any of our single antibody conjugates in our total-seq portfolio within the same format, put them together, and stain your sample. You just have to choose the markers. You don't have to worry about what is attached to them, because once you have a list of all those different barcodes, when you go to do the data processing at the end, you can figure out what was actually there on your cells. Okay, so now I will talk a little bit about cell hashing. So what is cell hashing? This is a way to barcode different samples in order to combine them into a single capture lane, so for 10x, and then demultiplex them during your analysis. So in this case, just as an example, we can have 3 different samples. We use 3 different hashtag oligos, HTOs. In this case, they're blue, pink, and green. 1, 2, 3. Once they are labeled and washed so that we have individually labeled samples, they can be combined, captured, and then demultiplexed later on in your data analysis. How do we accomplish this? So our total-seq hashtags target ubiquitous proteins. In human, they're CD298 and beta-2 microglobulin, and in mouse, they're CD45 and H2 MHC class 1. Important for mouse, if you're working with a strain of mouse that maybe has a different MHC class 1 gene, make sure you determine whether the clone that we're using in here will work. Because it is both CD45 and H2 MHC class 1, if you don't have coverage of both, they still have the other one, so you can still get decent labeling. It still should cover most of them, although we're then relying on CD45, so lymphoid-like cells. These conjugates are premixed and ready to use, so you don't have to worry about adding them or mixing them together. And it enables what I guess was called superloading of a 10x lane, so you can load more than the 10,000 cells, and allows doublet removal because you can discriminate based on presence of more than one hashtag and knowing that that's more than one cell that was present in a oil droplet. Okay, so we have an application note on hashing and different ways you can go about hashing, and a video talking all about this on our website as well. And so those are resources that you can access. I think you can just kind of so BioLegend total-seq hashing, and it'll get you to those 2 things. In the app note, these 2 methods of hashing samples are described. So more simply, you can have hash, pool, and then stain, or hash, stain, and then pool. So in these, there's benefits and drawbacks to each of them. So if you're wanting to save on your total-seq reagent costs, you can hash your samples first and wash them, and then pool them together and then label with a larger pool of total-seq antibodies. It's a little bit longer hands-on time, and your cell viability may be compromised depending on the type of sample you're working with. If you have a fairly hardy sample, this is should be okay. If you have more cells that are a little bit more sensitive to handling and could die quicker, you might not want to do this particular protocol. Then we have the option where you can do your hashing and staining at the same time with your total-seq antibodies. So you'd have your sample, your total-seq antibody cocktail, and your hashtag, and those would be stained together, washed 3 times, and repeat for all the different samples that you have, then pool them together for the capture. This one's going to have reduced hands-on time, but higher total-seq reagent costs, because let's say, for instance, you're using universal cocktails and you need to use 1 per sample, then you're going to have to use 3 cocktails here, whereas in this one, you're only using 1. So just as a comparison, because there are other ways that you can do multiplexing, and this one is not related to the flex and barcoding multiplexing, but this is comparing the BioLegend total-seq A, B, and C antibody-based hashing to 2 lipid-based hashing systems. And so this was an independent group. We had nothing to do with this. We just found out a bit later on, which was kind of nice. It was good for us. And so what we see here is that antibody hashing on cells demonstrates better accuracy than lipid hashing. And so in cases, they either outperform or perform as well as the lipid-based multiplexing methods. Just depends on, I guess, what you're doing. So they did do single-cell RNA-seq and single nuclei RNA-seq with human and mouse samples. I think the one where we were performing more equally well, but not outperforming, was for single nuclei RNA-seq. Okay. So now we will talk a little bit about cocktails because I sort of mentioned them already. So let's kind of dive in. All right, our pre-titrated cocktails. So these have a lot of benefits. Most that they are pre-titrated for optimal performance, and they're sold as a single reaction vial. You can label 500,000 cells per vial, which is one test. So they're sold in a pouch of 5 cocktails. And because they're lyophilized and individually vialed, you can reconstitute one at a time. You don't have to do them all at once. You can open the pouch and do one, and one this week and one 3 weeks from now, and it's still the same performance. Each antibody in these cocktails has been individually titrated using NGS as a readout to provide optimal discrimination of positive and negative cell populations. So what we did, I don't think I have a slide on it here. Oh, there it is. You can see a really tiny picture down here. But what we did is we actually labeled a sample with different hashtags. So the same donor, but then split it based with different hashtags and then different amounts of antibody, and ran them by NGS to actually see what the optimal concentration would be, so you can get discrimination of those positive and negative peaks. We did that for every single antibody that's in these cocktails and to figure out the optimal staining concentration. I will note that they were tested for human on PBMCs. So if you have a different cell type, there might be some differences in what you would see staining wise. So we have these for human and mouse in all 3 formats for universal cocktails, which these are the 100 plus or around 100, I guess. Total sig-B is a little smaller for mouse. And lots of different antibodies and isotype controls to help filter out events that you would like to exclude. They have excellent long-term stability in that lyophilized format. And it reduces inconsistency with pipetting and handling multiple vials and samples. Anyone who's done a 20+ color flow cytometry panel knows that that can be a lot with pipetting all kinds of different individual reagents. So this really would be a time savings to do the universal cocktail. We also have a myeloid cocktail. Myeloid's more myeloid specific for mouse in total sig-B format with 84 primary antibodies and 8 isotype controls. And then they are notably, while you might see looking at the actual price of the thing, a little bit of sticker shock, they are a significantly reduced cost compared to buying individual antibodies for a panel the size that you're looking at here. So why would we want to do this with hundreds and hundreds of different antibodies? Maybe not hundreds, 100 plus. So we initially made up a 9-plex panel, these T, B, and K panels in human. And so if you look at here, there's CD3, 4, 8, 11C, 14, 16, 19, CD45, and CD56. So that gives you all the major lineages, or several of them anyways. But what you can see is you just get the discrimination of, okay, T cells, CD4s, CD8s, your B cells, NK cells, monocytes, dendritic cells, other monocytes. But what's within that CD8 cluster there? And so when you add, we had a panel before we narrowed down our total sig-A universal cocktail, we had a panel that was 210 antibodies. And by adding that, by increasing the plexity of that panel, you're really able to take this CD8 cluster and make it into this many more, where you have naive cells, central memory, effector memory, T effector memory. We can look at gamma deltas, we can look at mates. So looking at more markers really allows you to uncover important cell information and gain a deeper understanding of the cells in your PBMCs or wherever your samples are coming. Okay, so a little bit more detail on the cocktails. So the human and mouse universal cocktails of 100 to 163 primary antibodies, mainly immune cell lineages and sublineages, some cellular activation markers, memory, exhaustion, signaling, chemotaxis. We also have the human TBNK. So if you do just want to look at those 9 primary antibodies or start there and add to them, because all of these cocktails, even though they're lyophilized, you can add antibodies to them. You could buy them in single vials, and we have guidance for what's called a spike-in, where you would reconstitute the antibody or the cocktail in a liquid containing, your additional 1, 2, 3, 4, 6 antibodies that you want to add to that cocktail. So if 9 is your speed, you want to add, I don't know, 10 more as opposed to going up to 100, then you can absolutely do that as well. As I mentioned, we have the mouse myeloid cocktail in total-seq B format. A and C are coming soon. And this is optimized for characterization of myeloid lineage cells, and it's recommended for use with enriched myeloid populations. So we actually tested it on spleens and bone marrow that had been depleted of T and B cells before looking at this. Because when you're capturing, if you think about it, 10 or 20,000 cells from a sample that are mainly T cells, when you try to go do those myeloid cells, if you haven't depleted the T and Bs out of there, then you're losing a lot of information. You're kind of getting, cutting down to a very small number of cells looking at those myeloid lineage cells. So it's nice to be able to, you do that depletion first, then you really get into characterizing what you have in terms of that myeloid population. And then this is for total-seq D, but this human heme-oncology cocktail, it's 42 antibodies. So if you are interested in a single-cell DNA-seq, we have a cocktail for that as well. So key takeaways here. These are lyophilized cocktails. They're in single-use tubes. They're pre-titrated using optimal concentration for staining, validated using NGS as a readout on PBMCs for human, splenocytes and bone marrow for mouse, and it will help to reduce inconsistency between handling samples in multiple vials and pipetting, reduce costs compared to individuals, and then because they have excellent stability and consistency across our lots, you can minimize variability between experiments, between labs, and during longitudinal studies. Okay, so with that, we'll switch a little bit gears to our intracellular conjugates. So this can enhance your studies using the chromium flex gene expression by adding intracellular targets. So for a long time, when CytSeq first came out, people said, this is great. I love the fact we can do surface proteins, but what about all my cytokines and stuff I want to look at? And so it was a long time coming because, there are some challenges and technical limitations by looking at intracellular markers. So surface staining live cells, that's no problem. That's okay. But when you want to stain fixed cells, then you can't do it, they're dead. So your viability is shot. But when you want to do surface and intracellular staining, they have to be fixed and then permeabilized, so they're dead and their membranes are compromised. So there's a number of different challenges here. Some things, the fixation can alter the epitopes of surface and intracellular proteins, which can lead to inaccurate staining. Unlike, formalin paraffin embedded, you can't use antigen retrieval to sort of reverse that fixation process. It's not feasible for cells in suspension. It's possible that you could have high background due to non-specific binding of antibodies. You could have higher false positives when you get antibodies stuck in here if they're not accurately washed out. And fixation can cause RNA degradation, which can affect the accuracy of transcriptomic data. So, when 10x kits first became available before Flex was a thing, definitely we're not doing that on fixed RNA. You had to make the decision of being able to do things with live cells. And so the advent of Flex, we are able to incorporate this into the workflow, which is really nice. So our protocol here for intracellular cytoplasmic protein staining simultaneously detects cell surface and cytoplasmic proteins with that gene expression. This is going to be able to enhance your cell characterization, looking at functional molecules, signaling pathways. Like I said, this works with the 10x Flex gene expression assay and chromium fixed RNA profiling kit for single plex samples. I think it is also compatible with their multiplexing system as well. You would do your surface staining here before fixation to improve epitope stability and the ability to do your immunophenotyping. And so a note here, total-seq B and C are supported with certain Flex applications. So consult 10x documentation. It kind of gives you a decision tree based on what you're planning to do, which will tell you which total-seq format to use. But you can also contact us, myself, or our tech serve team, and we'll be able to help you with the choice of what antibodies and what format to use. So the workflow sort of looks like this. You have your single cell suspension, you assess your viability and count. That viability obviously is going to be noted once you do the fixation, the cells are dead. So you have to count and do the viability upstream of that fixation step. You dilute your cells in an appropriate volume. This is dependent on the antibody pool or cocktail that you're using. Then you would do your blocking for self-surface Fc receptors. And then stain your cells with your total-seq cell surface antibody pool or cocktail. So this is just a surface step. If you have hashtags, you can have that as well. If you're doing a different cocktail, you have a different staining volume, but that's all listed out in the protocol. You would then wash your cells and then fix them. And at that point, there is like a stopping point. You quench the fixation. There's, I think 10x said you can put the cells in the freezer and hang on to them for a while. They can also hang out overnight. In our case here, we're recommending you can store them overnight, or I think up to a week in Cytolast buffer, which is something we sell. Then you would do your permeabilization and block those fixed cells, and then stain your cells with total-seq intracellular antibodies. Wash and then proceed to your 10x protocol to continue. And then make sure you're using the right guide that they have provide. So just an example of some data that we gathered using this protocol, where we have surface and intracellular. So we had a single donor of human PBMCs. We had in our first sample, total PBMCs, no manipulation, and they were unstimulated. And then we had our CD4 enriched population, and then that was stimulated with PMA and ionomycin and Berfeldin A for 6 hours, because anyone knows if you do intracellular flow staining, you have to do that step to kind of get the cells to produce more cytokine and trap it in the cells so that you can actually see it, so it doesn't escape. And then we had CD14 enriched cells that we stimulated with GM-CSF and LPS, and this R848 stimulant with Berfeldin A again for 6 hours. So after those stimulations happened, we harvested the cells, washed and count, aliquoted 1 million of each of the cells per condition into our staining tubes, FC blocked, stained with hashtags 1, 2, 3, and universal cocktail for each of them, and washed 3 times. Then we pooled all of the cells, so 500,000 per hashtag and per cocktail that was labeled there per condition. Washed, fixed, did the perm block, and then stained with our total-seq intracellular antibodies, because the intracellular antibodies could be applied to the whole mix of them. I mean, technically, we could have used one cocktail for all of them if we had hashed them and washed prior, but this was the other method that we talked about here. So we had our self-surface cocktail, total-seq B universal cocktail with 143 antibodies, 28 intracellular antibodies from our catalog, which was all the ones that we had at the time. They include cytokines, interferons, granzymes, perforins, chemokines, intracellular CD20, CD68, intracellular CTLA4. So there's a variety in there to see some different things. So here we have a cluster projection of our CD14 positive cells. So we have some monocytes up here, transitioning monocytes, non-classical monocytes, those are the classical ones. And then in the pink cloud, sorry, that's the CDC1 and CDC2. So this was enriched for the CD14 population. And so here we can see, we can compare our IL-6 antibody-derived tag and IL-6 RNA. And so they're pretty concordant, showing where they would be in the different clusters of the cells. And then same thing here with our IL-12 P40 ADT and IL-12 B RNA, and they're pretty concordant as well. This may not be the case in terms of where you would see exactly the same, because like we said, RNA and protein are not always regulated the same. So we could see some differences there, but it's nice to show that we can actually see both the ADT and the RNA. RNA hasn't been totally destroyed. So we got both types of data out of this set of experiments. Okay, so I'm going to switch gears away from RNA for a little bit to talk about a new kit that we just recently released called our total-seq A Phenocyte Kit. And this allows you to do high-dimensional single-cell protein profiling. So what is total-seq Phenocyte? This is an assay that uses instrument-free cell partitioning. So we use the Scale Biosciences plate-based single-cell quantum barcoding technology to enable this. It's high throughput and scalable because you can do 200,000 to 1.2 million cells. So you can do small and large scale studies. We found it to have really high sensitivity and low background, capable of detecting rare populations as low as 1 in 10,000 cells. This is going to utilize one of our pre-titrated, optimized antibody cocktails. So in this case, our total-seq A universal human cocktail. And with that, you're able to do ultra high parameter protein multiplexing. So you can't touch 160 parameters with flow cytometry at this point, but here you can. And so it simultaneously measures hundreds of proteins, so you can get really comprehensive profiling. And the data processing analysis is relatively straightforward and enables the use of our multi-omics analysis software to do that. So if you don't know what the Scale Biosciences kit is, or the quantum barcoding plate, I'll describe it a little bit here. So again, this is a single cell protein only assay, so we're not looking at RNA. We're using the TotalSeq A universal cocktail here. This kit's going to come in two formats, either a 200,000 cell capture, which would give you one TotalSeq A cocktail, or 1.2 million cell capture, which will give you 6 TotalSeq A cocktails. So again, this is the same exact cocktail that we talked about a little bit earlier. Nothing's different about it in this kit. So again, with this instrument-free plate-based approach, this plate contains a really cool technology that allows you to have the cells sort of fall into individual microwells of a 96-well plate, and then allows you to do indexing with I7 primers and some dilution of cells. So the sizes of our kits come, like as I said, come in 200,000 cells or 1.2 million cells based on how many cells you want to recover. So if, you're going to use actually more than that in your staining process. And again, like I said, one cocktail comes with the 200K kit and 6 cocktails with the 1.2 million kit. In the 200K kit, you're going to use 2 columns of this plate, and in the 1.2 million cell, you're going to use all the plate. So in columns, 6 columns across the plate. So what does this workflow look like? You have your sample staining. So similar to our other processes with our universal cocktails, you're going to label your cells all together with your TotalSeq cocktail. You can use hashing to multiplex samples in this case. But in this method, we would want you to do, depending on the size of the kit you bought, hashing your samples separately and then pooling them together and then staining with the antibody cocktail. Then you'll take your labeled cells and you'll do some indexing with provided I7 indices from the kit. And so that will end up, you'll have like, if you use the 1.2, or sorry, this 200,000 kit, you'll have like 16 different tubes. They each have a different I7, and you're gonna just aliquot your mix of cells across all of those different tubes. Then after those are labeled, they will be mixed all together and then loaded onto this plate. Like I said, this plate has this little micro well in it, and it uses these gel beads that won't fit into the microwells until you spin the plate down in a centrifuge. So the cells have all settled down to the bottom of the wells, but the beads sit on top, kind of like an ice cream cone. Then you go and put it in centrifuge, and then the bead like poof, squishes down into the bottom. And then the bead barcode will be applied to the cells that already have an I7 index. And so even if there's more than one cell in a well, you're going to have hopefully unique combinations of, like, let's say, the I7 that was red plus the bead barcode, and then the I7 that was yellow plus the bead barcode, and then you have 2 different cells. So once labeled, all this stuff gets dumped out of the plate using a fancy, cool little funnel, gets put into a reaction to fill the gaps between the different pieces of the barcode. The samples are then indexed with an I5 primer, and then you construct libraries, QC them, sequence them, and then you go to our multi-omics analysis software to do your data analysis. So the data analysis side of things, you get BCL files out of the sequencing. We recommend using BCL convert for BCL to FASTQ conversion. And then the FASTQ to count processing is done using our software. Converse to site-seq analysis, you can use our multi-omics analysis software for that. But the count processing is done ahead of time, and you're uploading count files to our software. In this case, you're uploading FASTQs, and then we will do the conversion to counts in our software. The upload can be either local if you have the files, or we can do a connection into BaseSpace as well, so that we can just get them from there, as opposed to you having to download the many, many, many gigabytes that are FASTQs for this experiment. Then we'll basically enable some flow-like gating using mass, and then you can do visualizations, dimensionality reduction, cross-sample comparisons. So we're kind of looking at this part here with our multi-omics analysis software. this initial primary data processing is done with Illumina's BCL convert. And then, the sequencing ahead of time was done with the Illumina sequencers. So what that data analysis looks like. So we have this free mass tool online. You can create an account and log in and start playing with data. It's in a web browser, so it doesn't really matter what kind of computer you're using, as long as it has a modern web browser. You upload those FASTQ files, like I said, using via local storage or base-based connection. And then we're going to get to do all kinds of different flow-like gating to really subset the different types of cells that you've used 160 antibodies to determine, or 150, sorry, antibodies to determine what exactly they are. You can look at different clustering algorithms and dimensionality reduction plots. And then that workspace can be shared with collaborators or our tech team so that we can kind of take a look at things that may or may not have happened when you were doing your experiment. So this is just a, whoa, timed me out. It's like, no, you can't be connected. It says it's connected here. Like, you've had an hour, you're done, lady. Well, I can't really show you what data visualization looks like when we have no visualization. So should I start drawing clouds on here? Sure. Yeah. Mhm. Yeah. Correct. Yes. Yeah. Yeah. So you get your matrix features and barcodes files, and then you put those are the counts, and then you use those to do your analysis. I'm about to talk about that. But I know it's kind of like when I put this together, I was like, okay, I'll talk about phenotype. I was like, then I have to go back and talk about mass again. That's fine, whatever. You'll just get to hear a lot about mass eventually. You get to see it. Not have signal. Present. Uh-oh, maybe I broke it. It broke. It's off. It was working up until now. I just turned the screen turned off. The screen just turned off. Yeah, just blanked out. Oh, there it is. The connection.
Yeah.
Oh,
Got USB-C.
oh, I do have that. That's on the other side. Ta-da! There we go. Monitor. No, it's need the displays. You need the settings for displays. Oh, there we go. Never mind. Got it.
I think.
Yep, I'll go and present.
Okay.
Thank you.
Yep.
There's a time limit on how long the HDMI cable wants to work on my computer. Okay, so there were some pretty clouds, but now we're going to talk about mass in the interest of time here. So what is mass? Now that I already told you that it existed for one workflow, I'll go back and talk about a little bit more about it here. So it's a free suite of tools for secondary analysis of quantified matrices of single-cell multi-omics data using total-seq products for protein detection. So it's pretty accessible regardless of your bioinformatics background. Again, like I said, accessible through your web browser. You can use this flow-like gating based on antibody staining. So I will note that you do have to have site-seq data to use this. So if it's just single-cell RNA-seq and there's no antibodies present, I don't think you can use it because we need to have 3 different files, one of which is your features file. And you can create these different kinds of dimensionality reduction plots, still again, share your workspace, and you can export your project in this H5AD format for use in other single cell analysis programs. So we have a little bit of like a workflow diagram here, but I'm going to go into that again, turned on its side here. It says there's no support in mass for FASTQs, not for this workflow, but for the other one, yes. And we can't make your count files in this portion of our mass. So that the recommended here is to take your FASTQs into count files using Cell Ranger. Then we would do our filtering and demultiplexing, and all of this stuff can happen in mass. And so there's other, obviously, alternatives that you can use for these things: demultiplexing or RNA normalization, clustering, data viz, quantification analysis, and data integration. But you'll note that, a lot of these tools, they're kind of aimed at bioinformaticians, people who kind of know what they're doing with this stuff. And so those, show up in here a lot of places. And so the unique thing we have here is that this cell typing with gating is unique to our workflow and our suite of tools. So I will go through each of these steps in a little bit more detail here. But here's just a couple representative plots and pictures of things that we would do in this workflow. And so we'll start with the filtering and demultiplexing step. So in our workflow, we start with a UMI-based filter that you can use on this knee plot to try to eliminate events that we wouldn't consider, empty cells or useless cells. Then we use our isotypes that are present in our cocktails to do some filtering. So if we have cells that have more than 2 isotypes bound to them, usually we kind of assume that those are dead. So they shouldn't have so many non-specific antibodies binding to them. It's usually dead cells. There's actually usually not that many in data sets I've seen that you get filtered out with the isotype filtering. We also have a mitochondrial RNA filter for our CiteSeq workflow. And so that helps us to filter out some of the dead cells that way. And then we do protein expression doublets. So cells that we propose are probably not real based on their expression of 2 typically disparate markers. So for example, you wouldn't expect something to be CD3 for a T cell and CD19 for a B cell positive. And so we would exclude the double positive events from there. So you'll see a series of these plots that are already set up with some markers in them to help you exclude them. This is like a red blood cell filter. So that one gets rid of anything that would be positive for red blood cell marker. And so then you can adjust these plots based on, if you want to be a little bit less or more or less conservative, you can actually adjust to pull a lot of these in if you think, no, I actually think those are real, or those are the weird cell I want to look at, to make sure that they're actually included. So this is all fully adjustable. Same with the isotype gates and the knee plots and the mito filter. Then we can do some filtering and demultiplexing with our hashtags. And so when hashtags are present in your sample, they're identified automatically. And you're presented a table here that shows you the proportion of the cells labeled with the different hashtags. You can adjust these gates if they aren't correct for any particular reason. But then you can see on a UMAP plot what is actually identified with each individual hashtag, so that you can say, okay, well, I will use these as my 1, 2, and 3 samples so that you can identify them for your analysis downstream. Then there's an RNA normalization step. There's 2 different things. We do a highly variable gene analysis and a normalization to the RNA counts that helps to normalize your data. And then you can work on cell typing and projection. So once that's all filtered, and I kind of, you wouldn't have the RNA steps for the phenocyte workflow, but then you can go through and do that lineage type gating, determine what kind of cell populations might be in there. You can also do a clustering algorithm based on expression of different cell surface markers, and then select populations in there, and then do projections on either gated cells or clustered cells to look at the different types of cells within your sample type. And then you can colorize these projections based on cell markers. So I think here they're super tiny, but I think that these are looking at either T cell, B cell markers, CD4, CD8, whatever they may happen to be. I just can't read any of them. They're way too small and too blurry. But those would correlate with you can map it out to what those different cell type clusters would be. So you can determine where those different proteins are expressed. Okay, so just to wrap up here, our TotalSeq portfolio enables doing site-seq with oligoconjugated antibodies, allowing you to characterize different transcriptomically similar cell populations and expanding those capabilities, improving cell phenotyping. We have lots of different formats compatible with the workflows here and different manufacturers' kits. We have our cell hashing reagents would allow you to identify multiplets and remove them, minimize variability and reduce batch effect if you're pooling samples. And then you can also pool smaller samples to reach your minimum cell number. So let's say you have, you want to do 10,000 cells captured, but you have a lot of rare cell populations that you want to be able to target within specific samples. So even if you're going to do a flow sort, you could hash beforehand, sort, and then you end up with, I don't know, 5,000 cells, but you could have those two pooled together then to get to your 10,000 for capture, for example. We have our cocktails with our pre-titrated antibodies, reducing cost per sample, saving time and some money depending on how you use them. The intracellular protocol compatible with different 10x products and our TotalSeq C and B antibodies. Our TotalSeq C intracellulars just started releasing last week, so those will be available for you to purchase as well. And our new TotalSeq PhenoCyte kit for high parameter single cell surface protein profiling by sequencing using the Scale Biosciences quantum barcoding technology. We have a nice landing page on all of our TotalSeq reagents, so you can kind of investigate each of these a little bit further. Lots and lots of resources online, but definitely as well contact myself or myself and I will direct you to the right place to help you answer any questions and figure out what you might need to do your essay. So thank you. I'll take any questions. Mhm. Yes. I see what you mean. From what I have seen, you still kind of see those like little blobs of the doublets or maybe even more than that. I haven't seen that those get bigger. I feel like they just get spread out over more of them. I mean, you'll definitely get more doublets than you would if you're not using them. So that'll definitely change that number of doublets. But I think it's more robust detection than just like, oh, that seems like there's probably their RNA quantity of 2 cells. So, yeah. Mm-hmm.
And also the antibody like
Mhm. Mhm.
is that
Yeah.
workflow like
Yes, and yes, case by case. So it depends on, if there's an option to use a different clone for the flow antibody, if it's the same marker. So let's say, I don't know, CD19, for example. If there's an option for them to use a different clone for the flow part and then their CD19 TotalSeq antibody, that's what we'd recommend first. If there's a different epitope or a different marker that might accomplish the same goal, same thing. So, if they say, okay, well, could I use CD20 for the flow sort and then CD19? I mean, I know that those are not, they have different functions, but, this is where I would encourage them to talk to us as well. The other thing is it could be possible to label with both at the same time, just as long as they're titrated properly. You're most likely not going to saturate every single epitope either way by flow or with TotalSeq. So, I mean, commonly people will want to do a flow sort with a universal cocktail, let's say. And so we can tell you all the clones are listed, so you know exactly what's in there. So if you're able to plan around it for flow, but sometimes it's not, right? You can't plan around it. And so in that case, we just say, okay, well, what I would do is maybe do a test by flow first with the specifics you're going to stain with for the sort, and come in with a different color, same clone, if it has to be the same clone. And do a flow experiment and just say, okay, if I label with like PE and APC CD19, same clone, and I get to script like both, right? If you lose one of them, then you know that you're kind of oversaturating with one. And it comes into play for titrations as well. Don't overblast your antibodies for sorting. If you're using a universal cocktail, you can't change the concentration that's in there. But then it's important to try to think about what you would do for your sort for flow. And the antibodies hold up for flow, that's fine. They can be in there. It's just a matter of like, figuring out the clones and staining protocol. So it is doable, just let's talk about it. So if you're doing universal cocktail, we say 500,000. It actually can go up higher than that. I think we have a note now that you can use 2 million. But you don't need 2 million. Minimally, we'd like to see 500,000. The reason for that is because you're going to wash the cells. And so if you say, okay, well, I only have 2,000 cells, this is going to be very difficult because you have to do the prescribed washes to remove those unbound antibodies. There is a way there's an instrument, Curiox, it's a laminar washing system. If you have really small cell population or sample size for cells, I would recommend something like that because it's going to be a lot gentler on the cells and you'll keep a lot more of them than doing washing with like centrifugation. I think we did have a protocol at some point on how to use Curiox for total seq labeling. So that can be shared as well. But if you make up your own panel, we would recommend 1 million cells. Again, because of washing, you can probably get away with 500,000, but it's mostly kind of that sweet spot of like flow cytometry, single 100 microliters, 1 million cells. That's kind of the standard. So, yeah. Anything else? Any Zoom question? No? All right. Well, I'll free you early. Thank you guys for having me. Appreciate it. Thanks for tolerating my HDMI mess up.